So it's great to see everyone today. Thank you all for joining us. Today's U-RIG is just going to be another fantastic one. We have two speakers that I'm privileged to say that I know a little bit, and that I've known both of them for a little bit now through CAIRIBU, and then also as colleagues. And I think that they're both just doing awesome and fantastic work, and I'm glad that we have the opportunity to share that and perhaps kind of spur conversation and collaboration. It just speaks to the power of this community. So our first speaker today is Dr. Kymora Scotland, who is Assistant Professor of Urology at UCLA Health and the Director of Endourology Research. She's going to be talking today about her research. And then our second talk today is going to be Dr. Jashkaran Gadhvi, who is a postdoctoral fellow in Doug Strand's lab at UT Southwestern Department of Urology. So I'll go ahead and give the floor over to Dr. Scotland, and we'll go ahead and get started with her talk.
Fantastic. Thank you so much, Mike. So before I even start, I want to put in a little bit of a plug. So I am involved with the SIIU, which is the Society for Infection and Inflammation in Urology. We have a meeting every year at the AUA. And during that meeting, we have a podium session. And so we are currently having our call for abstracts. So, if there's an abstract that you couldn't get into the AUA or the work wasn't done by the deadline, and have some interesting work that you'd love to discuss at the AUA, please submit your abstract. You can send it to me, and my email address will be there, or you can send it to siiuabstracts@gmail.com. And we look forward to reading about your work. Okay. So on that note, I will get started. All right. So as Mike mentioned, my name is Kymora Scotland. I'm an assistant professor now at the Department of Urology at UCLA, and I'm an endourologist by training, and I continue to do a lot of my work in endourology, which means I'm very interested in kidney stones and some other minimally invasive sort of treatments for kidney stones and other diseases. But this talk is really going to be focused a lot on the role that bacteria may play in kidney stone formation. And part of that is because we don't really know how kidney stones form. We do know that it is multifactorial, and we know that it is something that starts at the crystal level. Now, the most commonly found stone is a calcium oxalate stone. And really, it's something that has an increasing prevalence in the last decade or two, affecting about 10 to 11% of Americans annually. So, not a small number of people. As I mentioned, it's multifactorial, and there are any number of things that we think can play a role in stone formation, including diet, issues with immunity, the presence of heavy metals, genetics, hormones, and potentially bacteria. Now, traditionally, people have thought that bacteria plays a role with respect to struvite kidney stones. I mentioned that calcium oxalate stones are the most common stones. There are some others, and a very small percentage of patients have these struvite stones, which are known as quote unquote infection stones. And they are associated with the direct action of certain types of bacteria, urease-producing bacteria, including Pseudomonas, Proteus, Klebsiella, et cetera. And then you've got apatite stones, which are a type of calcium phosphate stone, and they tend to be associated with struvite. Okay, so those two types of stones associated with infection, sure. And calcium oxalate stones have been, generally thought to be non-infectious. But what I've noticed as an endourologist is that several times I have patients with calcium oxalate stones who are infected. And so the question is, where is this infection coming from? Are they just people who have recurrent urinary tract infections? Well, it turns out that when we send these stones off for culture, they oftentimes will come back positive for bacteria. And so there are actually several groups who have done work looking at the stone cultures of calcium oxalate stones. And mind you, these are not necessarily in patients with UTIs. All right, some of these publications were done in patients who did not have a history of a urinary tract infection. The stone cultures were sent and bacteria were found. Some of the more common bacteria found were E. coli and Pseudomonas, which is important for my talk because those are the bacteria that I chose to work with. So I mentioned that bacteria stone cultures were found to be positive by multiple teams. Recent work by Bruce Falk, who, by the way, is a geologist, actually identified bacteria within struvite stone samples. Now, something else that they did was to look at calcium oxalate stones, and they didn't necessarily identify bacteria within those stones, but they, under a nanometer scale, analysis, were able to show really interesting layering to calcium oxalate stones. So that got me thinking, because what this looks like for all the world is something called a stromatolite stone, which I'll explain in a bit. But it led to my hypothesis that uropathogenic bacteria may actually promote calcium oxalate stone growth, and that they may do that using the same tools they use in biofilm formation. So why would I say that? I'm going to actually move forward to this and go back for a minute. So stromatolites are geologic stones. So that goes back to why Dr. Falk was interested in kidney stones. And they have these really interesting layering patterns. But what's important about the layering patterns is that they're actually as a result of microbial mat activity, which is basically bacteria using their biofilm forming activity to lay down these mats of organic infectious materials, and they cause minerals to form these organic/mineral layers and form these stones. So the first thing we did was to take calcium oxalate stones and do thin section bright field imaging and using some polarized imaging as well to look at the layering, and found, something that looks sort of similar. And I hope that you can see my arrow here. So you've got these mineral layers, and then you've got these really dark organic layers. And if you look close up, you start to see these sort of circular materials that look biogenic. I got really excited about that and started to ask whether or not kidney stones might be a type of stromatolite. And so we started working with colleagues at a company called Sigray, who have these really cool ideas about how to fuse different types of imaging modality. And so they have this three-dimensional X-ray tomography machine that they put together, which really incorporates X-ray microscopy and X-ray fluorescence microscopy. And we were able to do lots of work with them and show that there's this really interesting layering where you've got these organic layers and these mineral layers for all the world looking like a stromatolite. And so what we wanted to do at that point was to look at the substructure of the stones. Now, you remember that I mentioned that struvite stones are the known sort of layered stones, as a result of bacterial activity. And we've seen, and this is bright field here, we've seen this sort of similar layering, all right, and with struvites. And here again, using our high resolution imaging to sort of show here layers, whorls, and what we think are these areas that consist of actual porous structures. That's fine, okay? We know that bacteria form struvite stones, but what about these other stones? So we saw very similar layering in our calcium phosphate or calcium oxalate stones and our mixed stones. So that got me really excited, all right? And so I wanted to try to identify bacteria within the stones. And so we did. So we used scanning electron microscopy and looked at calcium oxalate stones, among others. We compared them to struvite and calcium phosphate stones as well. And what I want to show here is that this is not at the surface of the stone, because you might say, okay, fine, if people have UTIs, you've got the surface of the stone, which is completely available for bacteria to just sort of sit on and co-localize, and you can identify them that way. These are stones that we actually cut through. So this here is the internal structure of the stone. And you start to see these structures that look potentially like cocci and rods within the stone. And just, in the interest of time, we did a lot of work trying to identify these. We did lots of staining, and I have a little bit of some different data there, but rest assured that we did some staining at the SCM level as well. But we wanted to really convince people. And so we moved to photon as well as confocal microscopy, looking again at these stones. And again, this is not at the surface, this is within the structure of the stone. So here's a control, here's a stain for, there are a couple of stains here for lectin, which stains for, polysaccharides secreted by bacteria, for bacteria, and for some of the adhesins that bacteria will secrete. And what was really interesting here, I'll just move straight to 3, because this is sort of a zoomed in view of what we're seeing here, is that this is layering. So we're seeing this lectin within our organic layers, and that is where it looks like we're seeing the bacteria. And just to confirm, we have negative controls where we got zeolite, which is inorganic, non-biogenic, mined from a site in Germany. And you can see that there's just autofluorescence here. So very excited by this, but what does that mean for us? Okay, fine, there's bacteria within the stones, great. Maybe they just hang out there, and maybe as the stones grow, they layer and bacteria gets trapped within the layers. And so there wasn't really any clarity around what role bacteria might actually play in biomineralization of these stones. So then the next thing we wanted to do was to take a step back and go back to the in vitro level. I mentioned right at the beginning of my talk that it's really crystals that form stones. So we wanted to go back to the calcium oxalate crystal level and see what bacteria might do. Now, I should mention that this is not an idea that I alone have had. And so other people have thought about this. And typically what they do is to start with these pre-crystallized chunks of calcium oxalate, and then subject them to bacteria. And they've made some statements about bacteria sort of, hanging out at the surface of these crystals. I thought that wasn't necessarily the best way to prove to people that right from the beginning, bacteria plays a role. And so I was fortunate in being able to use some of my chemistry background. I did a lot of transition metal chemistry before I became a physician. And what we did was to start with de novo crystallization. And so we created our own crystals. We subjected those crystals to growth in the presence of bacteria, in this case, E. coli, but like I said, we did E. coli experiments and Pseudomonas. I'll go back and forth showing the data just in the interest of time. And then we grew these crystals in the presence of artificial urine. Now, I'll take a little step back here to acknowledge that lots of folks have some questions about artificial urine and would prefer that work is done with pooled urine. We've since done some work with pooled urine and will continue to do that moving forward. But I think there's a lot of benefit to using artificial urine and just sort of keeping a clean system. So we formed our crystals, we grew them in the presence of bacteria, and then we went back to using high resolution imaging to analyze the impact of that bacteria on these crystals. And so that was the major objective of the work that we've done over the past 2 years now. So the first thing we wanted to do once we formed our crystals was to see if bacteria actually localizes to them, and we did. So here, this is Pseudomonas, and you can see with the red arrows, these rods that are sort of sitting, they're GFP labeled, they're sitting on our calcium oxalate crystals. And we did confirm that these are calcium oxalate crystals using multiple modalities, which I'll go into a little bit later. So the first thing I wanted to do was to try to figure out if bacteria in the presence of these crystals made a difference at all. And so we wanted to look at the types of crystals formed. And in the presence of bacteria, our crystals were much larger, up to 280% larger. And again, Pseudomonas takes a little while to grow. So this is over a 10-day course as compared to controls. But also we saw that the formation kinetics were much faster in the crystals grown in the presence of Pseudomonas as compared to your control crystals. So, up to 2.5 times as fast. And so then we wanted to do a little bit more in vitro work utilizing a 2D model. And so we wanted to really look at the crystals, look at their morphology, and compare. The first thing we did, obviously, was to confirm that these things were in fact calcium oxalate. And so we used, beginning with Raman spectroscopy, like I said, we used a series of different modalities. So here you see your typical diamond-shaped calcium oxalate crystal. I should mention here and take a step back, there are about 3 or 4 different shapes that you can see with calcium oxalate. And even with calcium oxalate monohydrates. But then we started to see these hexagonal shapes, which you can see with calcium oxalate monohydrate, but are certainly much more uncommon as compared to your regular calcium oxalate. And you see that they're larger. So that was really interesting for us. But again, scanning electron microscopy is a 2-dimensional imaging modality. And so, we did some elemental analysis again to confirm that what we think we see is what we see. But the question is, okay, so what? Fine, you see a little bit of a difference in shape. Maybe they're a little bit larger. What does that really mean, right? I just want to mention here that we see something similar with E. coli. The crystals are larger compared to controls, but they're not, like with Pseudomonas, they're not those hexagonal shapes. They are the diamond-shaped structures. Okay, they just happen to be larger and they grow a little bit faster. I'll talk a little bit later about this, but E. coli promotes, like Pseudomonas does, deposition as well as propagation of these crystals. So then we move to focused ion beam SEM. And what that allows is for us to do 3-dimensional imaging because the FibSem cuts through the structure. And so looking here at your control crystals, you cut through and you see that you have fully solid crystals, as would be expected. So what happens with the crystals that are, again, here are the controls on the left, what happens to these really large hexagonal crystals grown in the presence, in this case, of Pseudomonas? We start to see that there's this, again, here, this is sputter coating. This is not bacteria. We start to see that there's this open area within the central portion of the crystals. You do have calcium oxalate layers, and again, trust me that we did check. But in the middle, there's this, what we're calling this etching. And so we wanted to sort of go through and just again confirm that these hollow in-center crystals really are calcium oxalate. We did a lot of elemental analysis. We did Raman. But what we see is this sort of layer by layer infrastructure of the Pseudomonas crystals. And then looking at the E. coli, grown crystals, we also see this sort of open area, almost like a collapse of the structure with the E. coli. And so what we're, right now proposing is that, there's something about the bacteria, that is allowing layering of these crystals in such a way that once we treat these crystals with all of the things that we need to do, sputter coating, et cetera, dehydration, that we need to do the FibSem, we have a collapse of this internal structure because it's empty. Or at least when I say empty, there's stuff in there that potentially is organic and not crystalline. Okay. But the other interesting thing is that we wanted to look not just at the formation of a single crystal, but what, crystals form stones, we have to look at the aggregation. And so what we noticed happened with control crystals is that, sure, there are small clumps of crystals. They typically sit side by side. And then you notice that that's what's sort of happening here. But when we look at the crystals grown in the presence of bacteria, there we have what we are calling this crystal twinning. These crystals are growing into each other. You can kind of see that here. And they form these much larger aggregates. You see these sorts of things. And that happens with E. coli and also with Pseudomonas. So again, what we're suggesting here is that something about the bacteria is allowing these crystals to really clump together in a much more tight fashion. And that may potentially be the reason why we see that increase in the formation kinetics. All right. So then the question now in the last few minutes that I have is, okay, well, what's this mechanism? And we thought there might be three main ways. One is that maybe bacteria just alters the pH, okay? And that change in pH allows an increase in the aggregation. Or maybe bacterial secretions are involved. We know that bacteria in the process of forming biofilm, okay, secretes all of these adhesins, but also has these appendages, okay, flagella, pili, that sort of thing. Or maybe the bacteria just in general adhere to the formed crystal and act as nuclei for these new crystals. So I'll go very quickly through pH because we found that there's not really a very big difference in terms of what the bacteria does in terms of changes to pH and the crystals. But we were able to show that there's a range at which bacteria works really, really well, and that really corresponds to the range that we would typically see in urine. So the next question is what happens with secretions. And so we did some work looking at what does bacteria secrete. First of all is EPS, right? Extra polymeric substances, as well as DNA, extracellular DNA. So we stain for DNA. We again use this lectin to look for our EPS and a biofilm stain. And we were able to actually identify biofilm, right, at the crystalline level. We also saw that just briefly at the stone level. I don't have time to show that data today. And what we saw was that, as compared to just, your typical media for Pseudomonas in this case, we see an increase in this, in artificial urine, okay, in the secretion of these substances. All right. So then the next thing we wanted to do was to really look just at the bacterial secretions and whether they affected crystal growth. And we wanted to do that under a system of flow. So we started off and we developed our own microfluidic system. And we were able to show that there was an increase in crystal aggregation as we increase the concentration of the bacterial secretions. Okay, and we quantified that here in terms, again, of number and size. All right. And then we looked at formation kinetics and saw very similar to what we saw just in the presence of crystals without a flow system, that there was an increase in formation kinetics, an increase in crystal size. Okay, so all really consistent with the work that we're doing. And then we started to look at EPS and eDNA. I'll show you eDNA data here, and saw that eDNA actually just on its own promotes calcium oxalate crystallization. All right, and then you can see that here as we increase concentration. So as we increase the bacterial DNA, you go from just a couple of crystals here and there to really what looks like a mat of crystals. So very quickly done. Again, we confirmed that these are our crystals. We use EDX this time. We also use Raman spectroscopy to confirm here. Okay, so all of these crystals are calcium oxalate and they are calcium oxalate monohydrate. All right. I mentioned again the effect of eDNA on calcium oxalate formation, and just wanted to show not just the crystallization, but this aggregation is what we see happening here. All right. And so in the last couple of minutes that I have, I'll talk about what our conclusions are and what we've begun to do since this work has finished up. So first, we see that not just bacteria, but biofilm forming bacteria alter calcium oxalate crystal morphology. And we're working actually now with Henry Schreiber on what I think is a really exciting project, looking at, because what he and his team have been able to do is to, from kidney stones, extract bacteria, lots and lots of bacteria, that my team is now looking at, because we are going to evaluate whether these biofilm forming bacteria are the ones really driving the stone formation, and whether other non-biofilm forming bacteria are playing a role as well, or they're just harmless bystanders. So that helps us to sort of zero in on who are the main players here. We noticed that bacteria, both Pseudomonas and E. coli, increase crystal aggregation, and so we're doing some more work and accelerate stone growth, we think, right? So crystal aggregation, we're going to do some more work looking at this in flow settings using a 3D kidney model system that we've developed, which I'll show you in a minute. And then we're really excited about accelerated stone growth. So we are in the process of developing a system where we're going to look at actual stones, not just crystals, and look at the effect of bacteria there. I mentioned we just developed this microfluidic model where we've got renal epithelial cells in our flow cell. We're going to be able to have this flow at physiologic flow levels, where we're going to have our calcium, oxalate, as well as bacteria being able to be injected into the system. We've done some preliminary work already. Our model works, and we've been able to grow crystals in there. So now our team is going to be doing some much more involved experiments looking at what happens in the presence of bacteria. And of course, none of this was done without the work of multiple collaborators, Gerard Wong, who's a bioengineer, and William Schmidt, who is his PhD student. William's done a lot of the work I haven't really shown as much of, but some of the work initially just looking at the confocal stonework. Neil Lin and Jerry Chen, who's his PhD student, are they're helping us with some of the modeling. And a lot of our external collaborators, the FIBSEM was done by Ken Shen and Jawi Lee, who's her PhD student over in Illinois. Henry, I mentioned. Aaron Celestian is our geologist who we work with at the Natural History Museum here in LA. And we do a lot of the Raman work with him. George O'Toole, who needs no introduction, obviously. And we've been doing a lot of work with him. He's been very kind and has given us multiple Pseudomonas mutants. Jeff Gelb and Wen Bing with SIGRE, and my lab members, including my former postdoc and my grad students and medical and undergraduate students. And I will end here.
That was just an awesome display of data, Dr. Scotland. I was just really interested in this connection between, like, I mean, almost like biogeochemistry and biology, and this fascinating layering effect that you observe. I have tons of questions, but I'd like to open up questions to our audience members. Go ahead, just raise your hand. I can call on you. If you have any questions, we can go ahead and get started. So while you might be thinking of questions, I certainly have some. These communities that are living in these stones certainly are exposed to a very interesting biochemical niche in terms of a nutrient landscape, right? And I was just wondering if you could, what's your thoughts on, like, what are they eating? What are they metabolizing? Oxalate is certainly a really poor carbon source, and I'm not even sure they have, what is it, oxalate decarboxylase and oxidase that make formate, which is horrible as a carbon source. So what are you thinking?
What are they working on? Exactly. So that's some of the work that we're doing now to try to get a better sense. Initially, I had all of these sort of, off the wall ideas about siderophores and whether this was a circadian rhythm sort of thing where bacteria, because you have all these, layers, right? It turns out that some of these layers grow much faster than 24 hours. And it's not just siderophores that are working, but urine is a really rich substance. And so what I suspect is happening is that they're pulling from the urine. But so I don't have a great answer for you yet, but rest assured we're working on that, trying to figure out, and that's part of the reason why I think that biofilm forming bacteria are going to be the main players here, because this is what happens when bacteria form biofilm on anything else. They don't have necessarily, right, all of these nutrients to work on. Biofilm that, when they form layers on Foley catheters and stents, for instance, and artificial sphincters. And it's the same sort of environment. And so I suspect that, if we're lucky enough to be able to find something meaningful here, that will be helpful for, many other people who are looking at foreign bodies in the urinary system, et cetera, et cetera.
I think it's absolutely fascinating. I see a question from Alex Velasco from Stanford. How's it going?
Hey, good. Hey, Michael.
Yeah.
Hey, Dr. Scotland. Great
Hey.
talk. It was really interesting to see your data on the eDNA and how that had a very significant effect on the crystal formation. And we know there's some relation also to the biofilm formation. What are your thoughts on sort of mechanistically why eDNA would have that
Mm,
effect? Do you think it's mainly like, I don't know, the charge of the DNA that's a driver, or
yeah.
yeah, what are your thoughts
I,
there?
I think it's a charge phenomenon. I really do. I think you've got, all of these calcium ions just sitting around, waiting, right, to react. And you've got, a lot stand up. And again, that is why we came up with the sort of three potential mechanisms that we came up with, because, bacteria on their own, right? Cell membranes have a lot of negative charges. But I think that's what we'll find with the DNA. We're doing some work right now with eDNA and single strand DNA to try to see.
Mm.
I think I'm gonna share again, because I think I included one slide that I think is just fascinating. Because we did some work with single stranded DNA. And this is what we found. I don't know if you can see here. This is what happens with single-stranded
Wow.
DNA, right? So even, like just crazier. And so I really think that it's going to be a charge phenomenon. We don't have the final word yet, but that's what we're working on.
Dr. Scotland, are you saying that you get a different stone formation with double-stranded versus single-stranded DNA?
I think I'm seeing something that's a little bit more complex.
Yeah, okay. Wow.
It's the same crystals, but there's
Yeah.
even more twinning. There's even more deposition. There's even more interaction between the crystals.
Wow. All right. I'm floor is open for any questions. I see there's one thing in the chat, not a question. All right.
Oh,
Well,
I'm
yes.
sorry. It sounds like some people did not see the slides. My
I
apologies.
I I checked in with a bunch of people also, and it seems like it was an isolated phenomenon.
Oh, okay, great.
Yes. They had a lot of great images. Well, thank you so much for sharing your research. I'm really excited to see where this goes, and I look forward to catching up with you at a conference about this.
Thank you so much. And I just want to apologize because I do have another talk that I need to give very soon. So I won't be able to stay for your fantastic talk, I'm sure. So I really want to apologize for that. But thank you everybody for your attention.
Thank you for joining us. Have a great day.
Thank you.
All right, and we're going to move on to the second talk of today, and it's going to be given by Dr. Jashkaran Gadhvi, who's a postdoctoral fellow in Doug Strand's lab in the Department of Urology at UT Southwestern. And he was also a PhD student in the lab that I'm from, Dr. Nicole Danisco's lab. So go ahead and take the floor, Josh.
Yeah, I just want to make sure you can see my screen.
Yes, it
Everything
looks
looks
great.
fine. Okay, perfect. Thank you for that introduction, Michael. Good afternoon. My name is Jashkaran. I'm a former graduate student from the UNESCO lab, currently a postdoc at the Strand lab. But today I'm going to be talking about my PhD research titled Understanding the Role of the Host Adaptive Immune Response to Recurrent Urinary Tract Infection. Now, what is a urinary tract infection? UTIs are initiated when bacteria climb up the urethra and invade the bladder, causing a lower UTI, also known as cystitis. If not treated at this stage with antibiotics, the bacteria can ascend the ureters and infect the kidneys to cause an upper UTI, also known as pyelonephritis. This is a more serious condition, and if left untreated, can lead to sepsis and could cause death. Uh, how is UTI studied in mice? Researchers have studied this vicious cycle of infection and inflammation in mice. To infect mice, millions of UPEC are actually injected into the bladder. This causes the cycle to start. This causes the invasion of UPEC followed by infection, which is caused by pilus-mediated attachment. Um, this causes inflammation followed by severe cystitis and exfoliation of the bladder epithelium. Because of this, the bladder undergoes urothelial remodeling, which sensitizes the bladder. This sensitive bladder is actually prone to reinfection. And similarly, even in a human bladder that has been infected, also becomes sensitive and is easy to reinfect. Now, our UTI pathobiology is not a one-sided conversation, it's a two-sided conversation between the pathogen as well as the host. In our lab, we've studied pathogens by understanding the colonization factors, toxin, and immune evasion mechanism uh, that are generated by the pathogens. But there are other factors from the host side of things as well. We know that there is a robust innate immune response. Today, we'll see the adaptive immune response. But let's start looking at the innate immune response first. So the host innate immune response to UTIs is very well defined actually in mice. Within an hour of an infection, there is a robust cytokine as well as chemokine response in the bladder, followed by rapid neutrophil infiltration. Neutrophils will kill the bacteria or lead to bladder exfoliation, meaning that the infected superficial cells will be removed or sloughed off. There is a rapid monocyte infiltration as well, which can differentiate either into macrophages or dendritic cells. Mast cells are also present in the bladder. They released IL-10, which is actually known to hinder adaptive immune response. And finally, there are various antimicrobial peptides like LL37 that work to kill the bacteria in the bladder. And so that's the innate immune response. However, the adaptive immune response has been very well studied in mice by Molly Ingersoll's lab, actually. They have studied this response in mice. They have a model for recurrent infection. In this model, they infect the mice with 10^7 CFUs of UPEC, and then let the mice resolve the infection for 3 to 4 weeks, and then challenge the mice again with 10^7 CFUs of UPEC, then assess the CFUs. This mimics the recurrence observed in women, but it's important to note here that the mice do resolve the infection by themselves, whereas the humans cannot do that unless they're given antibiotics. So what they've shown with this model is that the bacterial burden is actually reduced by 2 to 3 log difference in wild type mice 24 hours after a challenge infection compared to the bacterial burden observed 24 hours after a primary infection. So in the lab, they use this as a functional assay of memory response. They say that the naive mice, which do not have any immune memory of UTI, the CFUs are much higher. But then once they do have some kind of memory, the future infections, they'll have a memory response, and that hence there is a reduced intensity of the second infection. On the contrary, if the bacterial burden is not significantly reduced after this challenge infection, as is the case in RAC2 knockout mice, these mice do not have T cells or B cells. In this kind of infection, when there is no memory that is developed, they see that the levels remain the same after the infection. Next, they also used mu-M2 knockout mice. These are mice that lack mature B cells. And using this knockout mice, when they do the challenge infection in these mice, they see that they still see that the memory response is intact. There's still this difference between a primary and a secondary infection. This leads them to believe that B cells not being there are not actually helping with the memory response. So B cells are actually dispensable for the memory response in mice. So having ruled out the role for B cells, they decided to decipher which types of T cells are important for the partial protection that is observed here. They looked at CD4 as well as CD8 T cells together, and they find that when they are both depleted, the memory response is lost. But when they deplete either of them, they see that the memory response remains intact. So either CD4 or CD8 T cells are actually sufficient to generate immune memory to UTI in mice. The Mysore lab has also shown that aged mice spontaneously develop these lymphoid follicles, which they call it as bladder tertiary lymphoid tissues or BTLTs. These mice, these are not infected; they are just aged mice, and they found that the bladders contain these lymphoid follicles. These lymphoid follicles are filled with T cells and B cells. And previous work from our lab also has shown the presence of these lymphoid follicles in our patient cohort with recurrent UTI. So with these studies in mind, we set out to explore the landscape of the immune response to infection during a human recurrent UTI. The goals of the study was to elucidate the adaptive immune response in the bladder, to investigate the association between cytokine levels and disease outcomes, and lastly, to explore the humoral immune response in the bladder. To study this, we collected biopsy tissue samples from postmenopausal women suffering from recurrent UTIs, with the help of our collaborator here at UT Southwestern. These patients actually elect to perform a surgery known as CFT or cystoscopy with fulguration of the trigonitis. This is a procedure which is the last resort for treatment for a lot of patients. Once they're enrolled into the study, they go for 3 weeks of antibiotic treatment followed by 1 week of no treatment. And then on the day of the fulguration, the urologist enters the bladder surgically, and with the help of a laser, they obliterate the epithelium and removes everything that is in the bladder. But before they actually do that, they collect 2 biopsies, one from a visibly non-inflamed region, which we call it moving forward as control, and one from a visibly inflamed region, which we call it as an inflamed region. It is important to note, however, that the control region is more like an adjacent region to the infection. It's not a true control. But yeah, once these tissues are collected and processed, we fix them, embed them in paraffin, and perform various histological procedures to look for immune markers. So first thing, what we found was in the control region, we don't see many lymphoid follicles. However, in the inflamed region, as you can see, we saw multiple of these follicles. These follicles are filled with CD3 positive T cells and CD20 positive B cells. We also found macrophages, but not a lot of them. When we zoom into these follicles, we actually see that, yeah, they are definitely filled with T cells as well as a lot of B cells. Next, we wanted to identify what kinds of T cells are present through immunofluorescence confocal microscopy. And using IF, we looked at CD4 as well as CD8 positive T cells. And what we found was these follicles are filled with a bunch of these cells, as you can see in red and green. We quantified these cells as well. And what we found was the CD4 positive T cells are enriched in the inflamed region as compared to the control region, whereas no such enrichment was found for the CD8 positive T cells. So based on these results, we wanted to know why these CD4 T cells are higher than the CD8 T cells. Why are there so many CD4 T cells, and what are they doing? We wanted to check if there was a Th1 bias or a Th2 bias. What do I mean by that? So when a naive T cell actually undergoes differentiation, it can either be Th1 cell driven by the Tbed transcription factor, or it can be a Th2 cell driven by the GATA3 transcription factor. Th1 response is responsible for immune stimulation, and it's characterized by various cytokines like IL-2, interferon gamma, and TNF-beta. It's supposed to create a robust response to infection, and it aids in removing the infection. On the other hand, the Th2 response is responsible for wound healing as well as urothelial remodeling. It is characterized by various cytokines like IL-4, IL-6, IL-10, and as I said, it is responsible for wound healing and urothelial remodeling. It does in exchange to the downregulation of the adaptive immune response. So previous studies in mice have actually shown that the bladder prefers a Th2 response, which mediates epithelial repair in exchange of developing a robust adaptive immune response in the bladder. So based on those results in mice, we hypothesized that in our patient cohort as well, there might be a Th2 bias, which diminishes the ability of the bladder to provide long-term immunity in the bladder. So to test for this hypothesis, we measured urinary cytokines from 3 different patient cohorts. So when patients have a recurrent UTI, they go through this cycle of negative urine culture and a positive urine culture indicating UTI. So with the help of our collaborator, Dr. Zimmerman, at UT Southwestern, we collected urine from women who've never had a UTI. They've not had a history of UTI. They've never had an active UTI. The second cohort of patients was women who have a history of UTI but currently are in this cycle. So they currently do not have an active UTI. And the third cohort was the relapse cohort, which these patients had a history of UTI and they are currently having an active UTI. And we also added our biopsy patient cohort to these analyses as well. And what we did was we performed the BioLegend's Legendplex assay to simultaneously detect and quantify 13 different cytokines. We looked at IL-2, which is a Th1 cytokine, and found that it was elevated in our relapsed group of patients. We found that IL-22, which is a Th17 cytokine, was elevated in our biopsied patient cohort. We also looked at various different Th2 cytokines, IL-6, IL-4, IL-10. But what we found was only IL-6 was elevated in our relapsed group of patients. IL-4, which is a very important Th2 cytokine, was not elevated in our patients at all. And lastly, IL-10, which is also again a Th2 cytokine, was only highly elevated in our biopsy patient cohort. So this led us to believe that at least in our patient cohort, we did not find either Th1 or a Th2 bias because both of those cytokines seem to be elevated. IL-2 as well as IL-6 seem to be elevated in our cohort. So next, when we performed immunofluorescence on these tissues and observed them side by side, we found that the T cells and the B cells are present in the follicle, and they seem to be interacting with each other. We wanted to test. We wanted to assess the plasma B cell activity during the human recurrent UTIs by measuring the secreted antibodies in the urine. So we performed ELISA to quantify the total IgG levels released in the urine. And what we found was patients currently having an infection, the relapse cohort, as well as our biopsy patient cohort, had elevated levels of IgGs. We also looked at specific Ig isotypings to detect which isotype was highly abundant. And we found that IgG1, IgG2, as well as IgG3 was highly elevated in our relapsed patient cohort. IgG3 was not elevated, but interestingly, we found that secretory IgA, which is a mucosal antibody, is released into the urine and was significantly elevated in our patient cohort. So with all of these results, all of these antibodies being produced, what are they doing in the urine? Why are they not able to clear out the infection? So I think future studies should actually focus on that. We hypothesize that these antibodies that are produced are either not specific, but they are inactive against the pathogen, and we need to test for this. So in conclusion, what we found was that we found lymphoid follicles filled with T cells and B cells which suggest an active adaptive immune response. We looked at the CD4 and the CD8 T cells. We found various different cytokines which were elevated in our relapsed patients as well as our biopsied patient cohort. We also detected IgG in the urine, which suggests an active humoral response. And patients which are currently infected have these higher levels of IgGs. And isotyping revealed different types of IgGs and IgA, which are higher in our patient cohort. So in the for future studies, we need to characterize the specific regulatory immune cell populations within these lymphoid follicles, like regulatory T cells using flow cytometry. We need to characterize those. It would be interesting to elucidate the functional role of these immune cell subsets that we've found and what are they doing there. It'll also be wise to investigate the role of cytokines as a potential diagnostic as well as prognostic markers for understanding recurrent UTIs. And lastly, we need to understand whether these specific antibodies that are produced, what are they doing, what are their targets, and what is their role in recurrent UTIs. So in summary, these studies' conclusions shed light on various aspects of the immune response, and future research goals should focus on understanding underlying mechanisms and therapeutic applications of these findings in the context of bladder health and disease management. With that, I would like to thank my lab members, and I'll take any questions if you have. Thank you.